The simultaneous measurement of leaf reflectance with chlorophyll fluorescence analysis using an already known mutant plant model facilitates the establishment of new reflectance parameters. The construction of new leaf reflectance parameters by our method allows real-time monitoring of changing plant behaviors under natural environmental conditions.
Before beginning an experiment, set up a measurement system as shown in the schematic with a sample stage, light source, PAM fluorometer, and spectral radiometer. Next, attach a 10 cm squared steel plate with a 1 cm diameter hole to the custom-made sample stage, and fit 2 thin fiber probes tightly together. Wrap the probes with plastic tape and use a holder to clip the taped probes onto the sample stage. Position the probes vertical to the sample stage and attach a biforked light guide made of glass fibers to the halogen light source. Then irradiate the sample stage from both directions at approximately 45-degree angles, adjusting the light source so that light uniformly illuminates the sample stage without casting shadows. For simultaneous measurement of leaf reflectance and chlorophyll fluorescence analysis setup, in a dark